Hello, everybody. So today's module is going to be a little bit different. I'm going to make sure to zoom in because I noticed on the other videos, it's pretty zoomed out. So today's module is a little bit different. So today's module is a full research paper workflow. So you've learned how to analyze one single image, but what takes up an actual experiment? So all of this is built off of the same research paper from before. You can see that I was having issues saving a CSV and Google Colab. It's actually my second time filming this video. I had some Jupyter Lab to Google Colab issues. So all of this data is from the paper I've referenced in some previous modules. Nanoparticle microglial interaction in the ischemic brain is modulated by injury, duration, and treatment. The first authors are Andrea and Rick, and this was my first opportunity to really apply my microglial morphology and pipeline in what was a very elementary version back then to microglial images from the brain. And so I'm going to recommend for most of you today that you just follow along and that you don't need to download all of this data and run this. For the next module, I will provide a CSV of all the results that we get today, but all of this data is quite a large file. And so if you are going to download it, I would be very, very cognizant of how much room you have on your computer in order to do the download. Or you can download to Google Drive, which would allow you more space and will allow you to import into Google Colab. But I would just be cognizant that you can totally just follow along today and understand what we're doing and that it is just a high throughput version of analyzing a single image in a way that'll let us actually analyze these results in the next module. If you would like to download all of the data, it's available on Zenodo. Dr. Nance and I believe a lot in open science, making our data sets open. We believe that the only real way to improve microglial quantification and analysis is by having a lot of data to build models off of. So it's online on Zenodo under this link up above, titled Fluorescent Microglial Images for Analyzing Morphological Changes Due to Injury Duration in the Ischemic Rat Brain. You can go ahead and download it here. It'll download all of the TIFF files, so those are our images, and it will also download this data descriptions Excel file that will describe what the file names of these images are and what you're seeing within the actual images. I've already gone ahead and uploaded all of these images into my current working directory for Google Colab, and I've actually already run the notebook since it takes quite a long time, less than 15 minutes, but more than just a couple minutes, and Google Colab in order to run all of these. And I didn't want to just sit here with you empty. So today I'm just going to walk through the notebook, walk through how we get these results. The next module I think is more exciting, in which we analyze those. So what are we looking at over here? So we're looking at a large set of TIFF files. So our confocal saves the images actually in an ND2 file, which is like a Nikon dimensionality format. That includes all of the image information, not compressed, as well as a lot of information about our confocal. I do actually work in ND2 now in my modern workflow, but I used to convert to TIFF first because I found that was easier to deal with back then. So TIFF is a non-compressed version of the images that still includes all the metadata, but does not include all of the necessary Nikon metadata in order to work on this exactly how we used to on the confocal. These numbers on the left, this 4-56-2, this is a number so that the slice type is actually anonymous. And this 40x references the type of magnification we use for this experiment. The entire experiment, we used 40x imaging. In my more recent work that we hope to submit by the end of the year or early next year, we actually do combine magnifications. And I have some work comparing magnifications into how you can use different magnifications to get different aspects of microglia. But if you're a cell researcher and you have a different magnification, it's totally okay to use this platform on that. I'm just showing you a workflow analysis for the 40x images that we have. So the purpose of this is to really go through this OGD severity paper analysis workflow. That was the nickname we had for the paper a while back, but it's the BioTM paper that I showed you and linked up here. I think it's a very good paper. It was my first experimental paper within the lab. I had a perspective before that. So as always, the first step we do is we import our necessary packages. None of these should be unfamiliar to you all, as we're doing the same process as before. We're just doing it to hundreds of images this time. I would even say that this experiment, one of my first experiments, is small on the number of images needed side. This process is still definitely runnable on my personal MacBook Pro. I don't need cloud computing for this, which is really nice as far as accessibility goes. But some of our newer experiments are pushing the boundaries of what you can do on a personal MacBook, although the new MacBooks are quite nice and I think they could be run on that. So just be cognizant of how many images exist in your experiment and how large their file sizes are, because that will really change things. So this time I'm going to initialize some variables. I'm not going to leave this as XYZ CSV. I'm going to name this CSV as OGD severity paper CSV. I'll just label it there. I'm initializing a folder location, and I'm initializing the old file type as TIFF and a new file type as a PNG. A PNG is a non-compressible file type, although we do lose some metadata that I do convert to during this process. I'm also going to define some treatments here. So normally you want to do a study blinded, but in this case, we're not actually going to change any variables image by image, nor are we even going to look at the images at all. I ran a small test set. I built this out. I know that it works. I save all the images. I can see what happens to them. So I'm just going to deblind them here for the computer for analysis later. So here I've created a list. You can tell that by the brackets of all of the codes for our different treatment groups, where we have the non-treated control. These are slices that were kept for the same length of time as our other groups, but nothing occurred to them. We have half an hour of OGD, so 0.5, and 1.5 hours of OGD. So this is when we deprive the slices of oxygen and glucose for half an hour and 1.5 hours. And we also have 3 hours of OGD. And then we have 2 treatment groups. So for one of the OGD 1.5 hours, we applied azithromycin to see if that would work as a treatment. That's shortened to AZ. And another one of the groups we shortened, we applied superoxide dismutase, SOD, for at the 3-hour time point. So now these are all non-anonymous. If you need more explanation about what that means, see, here's the 40, 50, 30, 40X in the brain region of the thalamus. And if we look at 40, 50, 13, we can tell that that's 3 hours of superoxide dismutase after OGD treatment group. Then I made a list of lists here, treatment group, by just listing out all of the groups. If you're following along, I'll go ahead and scroll over to the right there. And I stated Zoom was 40X. I do have more recent experiments that try more Zooms, and this should work no matter what Zoom you are at. However, I do recommend having at least a complete picture of a cell or a subcellular component if that is what you're quantifying. And then the only regions we're quantifying here is just 3, the cortex, the hippocampus, and the thalamus. You can do more, you can do different ones. That is irrelevant to this. It's just how you label the images that's important. And then here I created this image number NP range 1 to 6 because the max number of images we have in any region is 6. What do we mean by this? So typically when we design an experiment, we would like at least 3 images per each region and at least 3 slices per each pup so that we can have some replicates to run. And oftentimes we can get a few images per region depending on how large the region is. Here we're using pretty broad regions. In some of my experiments, I use sub-regions of each of these major components. And in that, you can't get nearly as many images. But we don't overlap any of the cells, and so that's what we have here. And then I'm going to create these file lists from treatment groups. So I created this non-treated control. I create originally all these initialized variables of empty lists. And then I have these for loops. Yes, this is not the most beautifully coded thing ever. If you are a software engineer out there and you want to help me turn all of this work that I've done into a beautiful code that works really, really nicely, I would love to talk to you. I would love to take up your time. But as a PhD student that was also doing experimental work, this was my first run of it. It's pretty clunky. It did work pretty well, and it's pretty fast for how many for loops I have. More recently, I do have something that's a little cleaner, but I'm not a software engineer. I would love to partner with people to make it better. So basically what it says is for all the slices that exist in that group, go to that regions list we created and go to that count we created and make a list of all the possible file names that we could have. And then do that for every single group. I'm going to change all of these to just AZ. I labeled them incorrectly before. Cool. Then I make a variable that is a list of all of those lists. And then if I run that, I can see the name of every possible image that we could have. Oh, I said range 1 to 6 earlier. Python starts counting at 0. So there's 5 images for each region. So even though it's 1 to 6, it's 5 numbers. Then I'm going to define this variable properties list. You can look up all the properties that you can define if you look up scikit image region props or region props table. This is just a huge number of quantified features of each of the labeled cells that we'll produce. I don't even analyze all of these, but if you would like to do some self-exploration or you're a little bit more familiar with some of these, some of them are more signal processing terms, go ahead and analyze them. We'd love to see what you do. And then this is the really chunky one. So this is a counter. There's better ways to do this. Basically, I count and then at the end, I increase the count and I keep running. So for all of the file lists for all of the files that exist in this treatment groups file list, for that name, try to look for the cell, look for the cell location, see if it's there. If it's there, read it in. Split it into the blue cell M and the green cell M, where the blue is our DAPI and the green is our IBA1 or microglia. Threshold the images according to Otsu thresh, binarize the image, remove the small objects, label the image, fill the holes, label it again, create an RGB label. These don't need to be in here for the sake of this code. Typically, I do have an extra step where I save all of these RGB levels where I create them. So if you want to do that, add a saving step in here. I did not just for the sake of time. So that's actually redundant here. Measure all of the properties in that label image, not necessarily this label image overlay, that's just for viewing. And at the beginning of j equals 0, create me a data frame. Otherwise, append this second data frame to it. If the file is not found, just continue. Sometimes a file isn't found, sometimes a cell goes bad or a slice is bad, and we don't have a ton of images, or the region, we can't get as many images without like a bubble or something like that. So that's why I have that exception. You'll see all these warnings here. This append method is going to no longer exist in the future. So that's a problem for future me, but not today me. So ignore those. And when you do that and you wait approximately 10 minutes, maybe a little bit less, it will run it and it will return this grades data frame for you. So this data frame is 36,000 rows, where each of those rows is a different cell. So that actually ran pretty quickly. For 10 minutes, you analyzed 36,000 cells and a ton of these different features such as area. These are those B-box features I talked about in yesterday's video that allowed us to create those red squares around our cells. Area where the centroids are, we get just a ton of features. 76 columns worth of features. Here, I just added an additional metadata that's useful for the paper, such as the sex of the pup and the age of the pup. This experiment only uses 14-day-old male pups, but in my more recent experiments, we do sex comparisons and age comparisons, and so this can help analysis down the line to be better. And I also add on the pup ID just for information. This is just a way of cleaning up names. That happens a lot. It's where I spend most of my time. And this is a way of defining the treatment group based off of the pup ID for each of the cells. That way analysis is a lot easier later on. Then we can see the data frame again, where if I go to the bottom here and I scroll all the way to the right, I can see that we've added on this feature of the male, the age, the pup ID, and the treatment that it's associated with. And then finally, I just go ahead and I save this CSV. So here I just want to save it to my Google Drive. And if you give it just a minute, it is going to go ahead and pop up. So I'll hang out here in a second. But it is this CSV that I will upload for you all for the next module. We're going to do just the basics of statistical analysis and data visualization of the actual results. And yep, that's everything. So I know it was pretty quick, but I hope that it was a follow through that you could understand the purpose of why I was doing what I was doing, which was taking this work from just one simple image to hundreds of images and over 30,000 cells. And again, that's on the smaller side. Our more recent experiments also have Z-stacks, so their images are much larger. We include both sexes. I analyze them multiple magnifications. There's a ton of really cool things you can do. And although I'm showing this applied to microglia, it is not necessarily specific to that cell type. So thanks everyone for joining me today. I hope that was useful.